نتابع أعمال المؤتمر مع
We resume the proceedings of the conference with the second session under the title of Future Studies and Medical Advances, chaired by Dr. Hawa Al-Mansouri, Deputy Medical Director, Imperial College London Diabetes Center, United Arab Emirates. Let's welcome her to the stage. Thank you very much. I welcome you all to the second session and allow me to transfer to English language.
Please ask everyone to turn down the volumes on your phones so that we don't disturb the speakers once it's their turn to speak. Please take a minute and make sure your phones are either turned down or on silent. Second, I'm going to stick very strictly to the timing, so forgive me if I cut off our speakers. I've already told them that's not because I'm rude, that's because I'm actually watching the time. Thank you very much. So our second subject or second session is very near and dear to my heart. It's related to both medicine and to genetics, both of which I was involved in. The first subject that we are going to address is genetic revolution and the emergence of personalized medicine. Very briefly, in 2001, it was publicized or published, the human genome was published, meaning that all of your genetic markers, everything was published actually in a magazine called Nature. I still remember in a lab that we actually cut out the magazine and we framed it on the wall. At the time, it included about 90% of the human genome, and later on, they filled in the blanks. So at the time, we didn't quite realize how useful this would be and how many applications we could possibly draw from this information in daily medical care or in the clinic. It seemed like a far cry from the bench, from the research bench, to the clinics of where doctors and patients interact. So allow me to introduce our first speaker, Dr. Sayed Jaradat, Director of Princess Haya Center for Biotechnology, Jordan University of Science and Technology, Hashemite Kingdom of Jordan. Dr. Sayed Jaradat holds a PhD in molecular biology from Wayne State University, USA. He also spent 3 years as a postdoctoral fellow at the National Institute of Health, where I happen to also rotate. Dr. Jaradat is the director of the Princess Haya Biotechnology Center, located at the Jordan University. He has maintained an active research portfolio, which includes defining DNA variations and mutations for many autosomal genetic diseases prevalent in Jordan, with a particular interest in recessive diseases. Please welcome him.
Bismillah ar-Rahman ar-Rahim.
In the name of Allah, thank you, Your Excellency, the head of the center, for the invitation. This invitation is wonderful as it comes in the year of Zayed, the late Zayed, may Allah rest his soul.
Could be translated and affected our life from bedside to clinic. So in 1953, that was a major breaking news, the discovery of the molecules of life, if you like to say. So that was in 1953. 50 years later, before than that, we learn what this DNA supposed to mean. It's a 4 base of nucleotide, nitrogen nucleotides, bind to deoxyribose sugars with a phosphate group, and they are complementary to each other, where guanine would bind to cytosine and adenine to thymine. So this is what your DNA is supposed to mean: nitrogen bases plus sugars plus phosphate group. How this make your life, how make you different from the others. So 50 years later, as Dr. Hawa mentioned, in 2003, the final genome draft was released in major publication, Nature and Science, and the 2 gentlemen, Francis Collin, the NIH directors, and Craig Finters from private sector, Celera, published both genome on the same time, and that was announced in a presidential setting, and this is how important to engage politicians in all of this active research going on. So this is your genome, ACTG, GCT, whatever it is. This sequence would make your life, 4 nucleotides repeatedly in every cells of yours, 3.2 million times. And this is what we call it reference genome. What reference genome? Whatever we are finding sequence today of a DNA, this is what we compare the current sequence with this reference genome in the databases. So this is, you look at chromatograph of a DNA sequence. Could be any of your DNA. And the technology used before, what we call it Sanger DNA sequencing, it took them for the first genome from 7 to 10 years to finish the first draft with a cost of over $1 billion. That was very costly job to do. But it's worth it? Yes. So 3.2 billion base pair in every cells of your body. You have trillions of cells in your body. So if you put those DNA and coil them, stretch them, that should be enough to take you back and forth from the earth to the sun over 600 times. Amazing. So what this DNA consists of, there is something we call a gene. The gene which make you happy, which build up your muscles, which make your eyes look good, and the same gene would give you diabetes or cancers or whatever it is. So the first mutations in the century described in our genome was the one causing thalassemia in the beta globin genes. So everybody of you have 2 copies of this gene, as well as have 2 copies of everything in your genome, one coming from your mother, the other one from your fathers. So that's what we call them alleles, or 2 copies of the genes. And these genes are located in chromosomes in our cells. As an example, beta globin genes cause thalassemia, located in chromosome 11. So the whole number of genes in our genome are scattered on all of these 23 chromosomes. So what this genome is consisting of, surprisingly, after the human genome draft release, just only 1.3% of our genome are what we call them exomes. Exomes are the one who could produce protein in the genes. But other than that, 50% of your genome is junk, we call it. Doesn't do functions yet. And another over 40%, we don't know what's the function of it. But there's a few portion, around 8%, they regulate your life. They regulate your genes. So as an example, that's what make us different from animals. If I look at my genome compared to a monkey genome, I am almost similar to a monkey and mouse with the same number of genes and with the same homology of the sequence. Almost 99% of my sequence are similar to a mouse. So these regulatory elements are important because apparently they are playing a role to make me different from a monkeys. If a gene is active, make me talking to you. This gene found to be in monkey inactive, so if you correct this gene in monkey, don't be surprised the monkey would walk by you and telling you marhaba. So technology move. Now, advanced technologies make things is happening in real life. 2 major breakthroughs happening in the last 10 years. One of them, what we call it, microarray technologies, where we could study 20,000 gene profile, how they are expressed in cancer cells compared to normal cells in one shot. And what you see, those red and green spots, suggest the differential expression between cancers and normal, as an example. At the same time, we could know variation between me and you with over 1 million variations using these technologies. So, on average, between me and any one of you, there are 3 million base difference in our genome. That's what makes my life sometimes miserable, and this is what could make your life happy. So these variations, we call them, are very important to design larger study where we can start looking in something we call genome association studies on these issues. So the other breaking news in 2007 and afterward, the next generation sequencing, الجيل الثاني من DNA sequencing. Remember, we used to say 10 years with over $1 billion sequence your genome. Today with these technologies, where they could use sequencing by synthesis, millions of fragments of your genome would be sequenced in parallel, massive parallel sequencings, assemble those, and put them against the reference genome. So on average, sequencing a whole genome, which took 10 years before, maximum it would take 3 to 7 days. That's amazing. With a cost over $1 million fold drop in the cost. In $3,000 probably maximum you could have your genome sequence. Why you need your genome sequence? That's a very important issue. So the issue, it's still debatable. There's many ethical issues around it. If I could sequence any one of your genome, I can tell you many things about your futures. I could tell you by age of 30 you would wake up and walking with Huntington disease because it's an age of onset disease. I could tell you by age of 60 you are at very high risk to develop Alzheimer's. I could tell you a lot what could your futures holding for you. So now back to the issue of this presentations. Many of you taking pills of codeine as example for headache relief. It's not working in you, but it's working in your friends next to you. So that's what pharmacogenetics is supposed to mean. In our genome, we have basically 2 genomes. One genome is the normal, and the other genome is, let's say, a cancer genome, when a gene mutated in your cancers. But interaction of both define what a precision medicine or personalized medicine is supposed to mean. So when you are giving a drugs, people are behave differently to this drug. Some of them will be good, some of them are not responding with toxicities. So these are the top 10 drugs on the markets. Look at the blue. Is not working in the blue, but not in the red. So on average, 1 in 3, 1 to 24 people, drug is not having efficacies. So one of the examples I want to show you on how our genome reacts to drugs. Warfarin is the most anticoagulant prescribed drug worldwide. It's an anticoagulant. Prevent clotting, so prevent myocardial heart issue. So this enzyme blocks something called VCOR C1 enzymes, these drugs. So this, when it blocks this, doesn't have enough reduced vitamins for clotting factors. So there's no clot formations. But when the dose is wrong, when these clotting factors inhibited, so you end up with a bleeding. So in every second in our hospitals, we have this issue. People are dying of bleeding at the time they are taking the drugs of anticoagulants. So interaction of looking at this gene is important when you look at those variations in this gene, define the exact dose for you, high, low, or not at all. This is the example of CYP2D6 as an example, as just I said. When you have a tablets of pain relief, it change to morphine, which is have an opioid activities. So some people doesn't activate this, so it doesn't benefit from that drug. Some others, they do. So now when it's come to cancers, precision medicine in cancer, this is melanoma, the skin cancers. A gene called BRAF mutated. When it's mutated, it's keep activating the whole pathway and the cell keep growing and dividing and become a cancer cells. So a drug like vimorabin block this mutation, and look at on the left, what's the difference make in 15 days treated with this antibody, the cancer is almost disappeared. So now, we cannot design drugs of every type of mutation or cancers. So people start looking at other type of cancers with the same mutation in skin cancers. Indeed, the same drug function on lung cancer with the same mutations. So look at this breast cancers. You have every minute now, you are sitting in this hall, a lot of DNA damage in your cells. But what doesn't make you feel sick or develop to cancers, as an example, because you have a repair system in your body. Fix this, minute by minute, second by second. So in this case, when a mutation like in BRCA1 causes these defects in cancer cell, another gene in the cancer cells fix this. So when we are inhibited the other gene, then the cell is die. So they develop this. So this is a study where we are looking at 10,000 patients of cancer, a whole genome sequence and exome sequence. So the conclusion of this study that there is a website now, a lot of data available on the drugs could match your mutation and match the type of cancers. So from all of the 10,000 people you look at it, there is at least 4,000 have an actionable plan. This means they can be put directly on the right drugs for this mutation for this type of cancer. You look at the lower panel, you see somebody with prostate cancers. And look at the size, how it shrink when they pick up the right drugs for this type of cancers, which is mentioned here. Diabetes. I just heard Dr. Jamal and Dr. Hawa speaking. How many in United Arab Emirates? She said 1 in 4. That's a very demanding health insurance issue. So now, what if a clinician like Dr. Hawa treats somebody with an neonatal diabetes for the rest of his life on insulin, but at the same time, he doesn't need insulin, this baby. Why? Because this baby, as an example, one of the neonatal diabetes form defect with a potassium channel-driven ATP. So if you define this mutation in this gene, all what you do, you give him a tablet of sulfonylureas instead of insulins, 1 daily, and you pass this defect, and the children or the child is normal for the rest of his life. Similarly, on a precision medicines, Moody type of diabetes, some patients, monogenetic, have a GCK deficiency, which is have high glucose when and fasting high glucose. So this type of patient doesn't really need insulin, doesn't need any treatment, just lifestyle a little bit and he's okay. So that's what makes a difference. We know exactly what caused these diseases. So one important thing, worldwide, they are studying these variation I told you about, which is if I have an A, you have a C. But if I have an A and you have a C, does it make a difference? So that's what make millions of variation studying to look at them in association with disease or in health. And for diabetes, over 100 genes or loci being characterized thus far, all of them explain only less than probably 20% of risk to develop diabetes. One of the gene interested is this gene, which they discovered in Mexican patients. When they look at the gene deficiency in hepatocyte, They look at markers similar to diabetic markers, increasing glycerols, triglycerides, إلى آخره. So this immediately, by understanding the function of this gene, they start developing an actions. Another gene, if you carry a mutation in this gene, reduce your risk by over 65%. That's a great advantage. Why? We all know why. Another issue I want to, you guys should not put this because that's make you nervous. On coronary artery disease, everybody go to the hospital, check his lipid profile on his way, start questioning, which is the good, LDL or HDL? I'm sure it's happened with 50% of yours. So thus far, there is no any genetic evidence to link HDL, the good one, as we call it, with the coronary artery disease. But there is other hundred of evidence link triglyceride and LDL. Look at those two genes from a genome-wide association studies. So one of the gene, PCSK9, now the function of this gene is degrading the low LDL receptors in hepatocytes. So what if you develop an antibody, a drug, could block this gene product from destroying LDL receptors? That's great. That's what they did. And reduced diabetes by 60%. But look at the frame line of developing a drug. It took almost 13 years with $1.4 billion. So I'm going to skip the other gene. This is the latest major technologies. CRISPR system. What is CRISPR? CRISPR is a system discovered in bacteria. It's like a vaccine system in bacteria. Infected with virus, it's killed the bacteria. So scientists took advantage of this CRISPRs by having stretch of DNA complementary to the gene you need to destroy. Like in this case, the PCSK9. They put it in liver cells. Successfully, they knock out the gene by 50%. So Dr. Jamal, next year, bring somebody to talk about this system because this will be the future revolutions in medicines. So less than 1 minute, worldwide people acknowledge the importance of Middle East in genetics because of the consanguinity issue to map quickly genes. But you look at the major databases and major science work worldwide, you don't see Middle East population representative. That's prevent us from benefiting from all of this discovery in genomes. But shortly there is some initiative in United Arab Emirates and a little bit advanced in Saudi Arabia on this. And I will be finished here. Thank you so much.
Thank you so much, Doctor, for being impeccably on time. Funny you mentioned the diabetics. I had the pleasure of telling a few of my patients that they never have to inject insulin again. And I have to say it was a very emotional moment. At first they don't believe me, and then they cry. So a lot of these genetic sequences do make a difference, and I'm proud to say that our center does run them. When we have a suspicion clinically that you are not the type of diabetes they told you you were, we run the genetic sequences and we find out for sure. So it really makes a difference in people's lives. Thank you for pointing that out. Second speaker today is going to talk to us about the aging communities and the need to redesign the healthcare systems. Now, we all know that as we get better and better at medicine, people live longer. Thankfully, we got rid of certain diseases. We've prolonged life. Certain continents like Europe have a bigger problem than others. They have an aging population. And in the Middle East and the UAE, this is occurring on a slower pace, but it's occurring as well. What does that mean for the healthcare system? What needs to be changed or what is changing, whether we like it or not? To present on this subject here is Dr. Nabil Najab. He's a geriatrics consultant, and after graduating in France in 1995, he returned to Beirut, where he became the founding president of the Lebanese Geriatric Society. He then joined the Dar al-Jaza al-Islamiyya Hospital, the largest specialized hospital for care of the elderly in Beirut. He rose through the ranks to become head of the geriatrics department and then medical director of the hospital. It brings me great pleasure to present him to you.
Thank you, Dr. Hawa. In fact, my talk will be in Arabic. My presentation, my slides are in English. بداية اسمحوا لي إنه
Please allow me to thank His Excellency Professor Jamal Sanad al-Swedi for honoring me by this invitation and to speak in front of you, especially for giving the concept of aging, an aging population, the importance that it really deserves within these major developments in technology. I believe that this lecture is the least technical one among all these presentations. This image that gathers several generations is no longer an exception. These are 6 generations that have become more present and persistent. I will start my discussion by defining the concept of aging. Then we will talk a little more about some statistics and figures and demographic changes. Then we will highlight the impact of aging and the process of aging itself on health and the ramifications on the healthcare sector in general. And I will speak about some of the challenges and some proposals as well. First and foremost, every individual, as he progresses in age, he will age definitely. So aging and reaching old age is a natural part of life. It starts gradually, and at the end, actually, it does not really begin when you reach an old age. It starts, according to international statistics, it starts at 65, but it may be raised to 70, but it does not really start at that point. They start at the late 20s. When you reach 30 years of age, you start aging, and the aging process starts, and the onset of aging begins at 30, and it leads to a change in the healthcare and in the health and the physical abilities, resources within the body, and he or she becomes more prone to diseases. Aging is not a disease, but it is the weakening of the body, and it is more closely related to the hereditary elements mentioned by Dr. Saeed. Moreover, they are linked to the environment and lifestyle. How do we live? How do we live? And this will tell us how we live today. Once we know how we live today, we know how will we age in the future. So, it is possible to speak about aging. However, it may not necessarily have a negative impact on one's health, because the time age, or the age in terms of time duration, is not really denoting the reality of the mind health or the physical health. The greatest example of this is the Nobel laureates whom we see in technology, physics, and literature. All of them are garnered by elderly citizens. So you can still be highly innovative while being an elderly individual, because you have gained experience. So in order to boost innovation, we need to pay attention to our health throughout our life. As for life expectancy at birth, it is being increased exponentially, to the point that by the mid of the past century, it was still growing slowly, but after the mid of the past century, it has been increasing in an exponential and unexpected rate. Now we are talking about young people who are 70 and 75 years old. So now we consider the age of 75, still they are young. So we are talking about the elderly of the elderly, who are beyond 85 and 90 years old. So this has become well known in all communities. And this is not really restricted to developed nations and industrial nations. Actually, the number of elderly population will double or triple. It will triple within the upcoming 40 years. So by the year 2010, we had the elderly population standing around 500 million people worldwide. Now, and within 30 or 40 years, the number will triple. And this is a massive increase, especially compared to the birth rates, which are witnessing a massive decline. So, whether we like it or not, we are moving towards a more aging community. So, if you look at our own region, we'll find that in this region, the number of elderly citizens is increasing exponentially. And let's take an example here. In Lebanon, the country where I reside and belong to, it was expected that the number of the elderly population would be 20% of the entire population, and now the figure has reached 30% of Lebanon to be an elderly population by the year 2050. And this increase is also is implemented and applies to the UAE, where 25% of the population will be an elderly population by 2050. This challenge has reshaped the concept of the aging pyramid. It's called pyramid because we see, usually traditionally, we see that the birth rates are a lot more than the elderly citizens, but in some cases the birth rate will reach, in 10 countries of the region, the number of the elderly population will exceed the number of birth rates, and this will create a number of challenges. And here we're saying that if I had known I was going to live this long, I would have taken better care of myself. What is the impact of aging on the body? As I've mentioned, it is a natural transformation that will come anyway when you grow old. White hair or gray hair is not a disease. Wrinkles is not a disease. The weakening of the kidneys or the heart muscle or even weakening bones, this is not a disease. It's a transformation that comes with aging. If we compare a 30 years old young man and the same young man, when he becomes 70 years old, he will still have 92% of the size of the brain, 70% of the renal capabilities, and 43% of the respiratory system. Does this mean that aging is a disease? Of course not. So this concept that I want to convey is that we are witnessing a reduction in the resources that we have, but it's not reaching the threshold of disease. So even if we age, we can age in a healthy manner. What matters the most is paying attention to our physical resources. The most important element here that exceeds by far physical changes are the social changes, which have a massive impact on psychological health and well-being and the activity level that a senior citizen can do in the community. After these some of these transformations, because some people represent aging as the age of many losses. The first one is retirement, losing your job. Secondly, widowhood, or losing your partner in life, and loneliness, losing your friends who are of the same age. And this shows that the first thing that the first paper they read in the newspaper, the obituary papers. So this loneliness that the elderly suffer from, it was really an exclusive situation in Western societies. However, now in our communities we are witnessing many elderly citizens who are living in loneliness. The whole change in the family. The elderly citizens who used to be the managers of the family and the leaders of the family now have become recipients. Now our children are taking decisions on their behalf. So all of these changes and social changes will definitely have a massive impact on the elderly citizens' psychology, and we have to combat them and face them in a different manner. When we age, we can reach a point where we become reaching a level of dysfunction, physical dysfunction. But we can still enjoy good health, as we see on the picture in the right, compared to the picture on the left. So, the most important message that I want to convey through this platform is that the picture to the right is just representing 10% only of the elderly citizens. Just only 10% of the elderly population that need help on a daily basis. So we should not represent aging as a reflection of the image on the left. 90% of the elderly citizens can become a massive economic driver in the economy and play an active role in the community. Why do we witness this change in the aging of our communities, all of our communities, if we can say so? The World Health Organization, WHO, attributes this change to the deaths. In the past, people died because of epidemics and diseases. Today, they are the NCDs, the non-communicable diseases, the chronic conditions that include arteries, hypertension, diabetes, cancer, heart failure, and so on. All of these diseases, we know that they are preventable diseases in a good manner and can be prevented in a good manner. That is why we need to focus on the prevention in this early stage, because we know for a fact that the elderly citizens, 60% of the elderly will have chronic diseases, and this will reflect on the cost that the community has to bear in terms of healthcare expenses. If an elderly citizen does not have any chronic disease, according to this study conducted by the American Hospital Association, it will cost $250. But if he has 5 chronic diseases, the cost will be 12 folds. So this shows how important it is to focus on this age group, to look into the resources that can reduce the impact of aging on the communities and individuals working and producing in this community. From this perspective, and in addition to the other chronic diseases, we have seen diseases that have risen into the spotlight, such as Alzheimer's and dementia. Dementia has become very widespread diseases. However, now we also have come to hear by protection from falling, because this is one of the most common problems that change the life of the elderly citizens. So the challenges are improving the quality of life to ensure that this age or this phase of life is an enjoyable phase for the elderly citizens, and also become a productive phase for the elderly. And in this context, we see the role of all the people working in the sector of improving the quality of life for the elderly citizens. And now we are talking about the number of multiple changes, which I'll highlight some of. The first one is the resources needed to continue ensuring that we have proper health care in the aging period. This funding is required, and we need to have specific researches to prove that it is far more viable to fund prevention of these diseases rather than fund the costs of treating them. So in the aging and the old age, for senior citizens, we do need to think of reducing the cost of healthcare in this period without undermining the quality of life. However, what we also face, especially in the MENA region, is the shortage of healthcare professionals. We know that in many hospitals, in many countries of the region, they use nurses, male and female, from overseas to overcome this challenge. The following challenge is the change in the family structure, because if they are looking after the elderly citizens, now they have changed according to many pressures. So the nuclear family, for many of the challenges that we're facing, especially in our region, there are many we're witnessing the immigration of many young people. So the concept of ensuring that the family is the first element of the community that looks after the elderly citizens, this has become a challenge, and communities should contribute to resolving this challenge. However, this requires facing a major challenge. That is why we do need national programs for looking after the elderly citizens within the community. The other challenge is that today there are many women who are working women and have become women holding jobs in our communities. However, the elderly women the elderly women today did not have jobs back in time. So this is another challenge in terms of the financial capabilities of those elderly women these days, especially in the MENA region, especially that the elderly citizens still have to work to earn a living and to cover the costs of healthcare services and social services. The biggest single most challenge is transforming the healthcare facilities and establishments from providing treatment to diseases to cure, or providing cures to prevention establishments at this age or this elderly age. This requires large teams that are highly specialized in this and dedicated to this domain. We are witnessing severe shortage in our countries. Some countries have not even witnessed the elderly citizens specialization, which has been introduced in Europe and the USA more than 50 years ago.
And نعم.
Therefore, we are in a stage in which we need to exert more effort, and there's also a huge shortage in the studies about aging, as well as shortage in the coordination between the specialized ministries. For example, is it a specialty of the social care ministry or the education ministry in order to educate the students about their condition when they age, or how to take care of the elderly and their families? Or is it the responsibility of the Ministry of Health or the Ministry of Labor? There is a lot of factors that are intermingled in this matter. First of all, we need to create preventive centers. We hear of the primary care centers and services. They are essential, yet they need to shed more light on the age that is more consuming of health, and among which is that we need to have a specialized team. For example, the aging doctor does not only tackle the disease, so they don't really just to treat a high blood pressure patient. He would ask him as well about the sleeping issues, the social problems, the family issues. We also need to tackle the point of independency, falls, mental health, and so on and so forth. A lot of things the physicians may actually pay attention to and we need to work on. We also need to shed the light on the importance of physical activeness of elderly people. There should be sports centers specialized for elder people in order to prevent them from falls and bone breaks and stuff like that. So also we need to shed light on the healthy eating. You shouldn't eat a lot of meat. You should eat more vegetables and fruit. This is not this is right in for youth, but this is not the case for elderly people. We ask them actually to eat more meat, since the muscle mass for elderly people is lost by 60%. So we need to have institutions and centers to create a sense of warm interaction between all people in order for them to interact with each other and not have trouble while living. And finally, we in this area we have a lot of something that we focus on here, which is the spiritual and religious part, which is after reaching an elderly age, we can enjoy it. And I couldn't find anything that is more a quote that is known for a very famous actress, which she says that to stay young is to live honestly and eat slowly and lie about your age. Because until this moment we can't turn back the time unless Dr. Said promised us to do so. Yet until this moment we should consider age only as a number. Thank you very much.
Fascinating conversation and presentation. Our final presenter unfortunately was not able to come in person. The presentation will be done via video. The subject of the presentation is, let me give you a scenario. Let's say, for example, that all the companies that make antibiotics, the medicine we use to fight infection, stop working, and you can't produce it anymore. What do we do? How do we treat infection? This is actually happening in reality, not that the companies can't produce them, but because the bacteria are becoming resistant or they're not dying when you give the patient the medication. The bacteria, the infection lives on. It doesn't care. So I will present to you now Dr. Mark Blaskovich, who's going to talk to us about this subject. Senior Research Chemist, Institute of Molecular Bioscience, University of Queensland, Australia. He's a Senior Research Chemist at the Centre in Australia. He's also the co-founder of the Community for Open Antimicrobial Drug Discovery, a global antibiotic discovery initiative in Australia. Please, welcome.
So in the last decade, there's been a lot of attention in the popular press about the rise of drug-resistant bacteria, otherwise known as superbugs. Now, while some of these headlines have been somewhat sensationalist, they do reflect a reality that the world is heading towards the post-antibiotic era, where common infections and minor injuries that have been able to treat for decades since the 1930s and the introduction of antibiotics can once again kill. And this threatens a real danger in that by 2050, it's predicted that we may have more than 10 million deaths per year globally caused by drug-resistant bacteria, which is more deaths than is currently caused by cancer. If we don't have antibiotics that are able to effectively treat infections, it's beyond just the simple types of infections that you think may cause death. So common medicines won't work anymore. So anti-cancer therapy often uses antibiotics to treat the immunocompromised patients. Things such as hip replacements, joint infections won't be possible anymore, and common surgery because antibiotics will no longer be effective. It's predicted that the economic costs of antimicrobial resistance will also be high by 2050, causing the GDP to drop by over 3.5%. So before we go any further, I just thought I'd point out what is antibiotic resistance. One thing that's key to point out is that resistance means that the bacteria are becoming resistant to the antibiotics. It's not your body that becomes resistant to the antibiotics. When the bacteria become resistant to the antibiotics, they're no longer able to be killed. So the bacteria can multiply and grow in the body and cause an infection, which leads to death. And how does this resistance occur? Well, when you have a high number of bacteria, a certain percentage of them are naturally resistant to antibiotics. And the antibiotics kill the ones that are susceptible, and as well as the good bacteria, but they stop the infection from growing. Resistant bacteria that remain are able to start growing and take over and become the predominant population. And once they become resistant, the bacteria can easily transfer the resistance between different bacteria. There are a number of causes that have led to this growth in antibiotic resistance. So one of the most common causes is the overprescription of antibiotics, so people taking them unnecessarily, people not following the proper course of prescription, so they underdose or expose the bacteria to sublethal concentrations of antibiotic, which fosters the growth of more resistance. A lot of antibiotics are unnecessarily used in agriculture. You can have poor infection control in hospitals and poor hygiene, which allows the spread of resistant bacteria. And finally, a lack of rapid diagnostic tests, which would allow you to tell very quickly whether you had a resistant strain or not. Now, antibiotic resistance is nothing new. Every single antibiotic has been introduced, from the introduction of penicillin in the 1940s up to the most recent antibiotics of ceftaroline. Resistance has been detected almost as soon as the antibiotic was introduced into the clinic. So what's been changing? Well, one of the main drivers of this increase in resistance is overuse of antibiotics. So this is a heat map showing regional variations of antibiotic prescriptions within England. Now, there's no reason why antibiotic prescription should vary from one place to another, but it just shows that in some places, doctors will overprescribe antibiotics. And even more striking is this demonstration of seasonal variation in antibiotic prescriptions in England, where in December, which is in the middle of winter, you get vastly more antibiotics prescribed, predominantly because this is cold and flu season. Now, colds and flus are caused by viruses, which aren't treatable with antibiotics. And so one of the main reasons is inappropriate prescription of antibiotics. And this just, you expose the bacteria to more and more antibiotics at sublethal concentrations, and it fosters the growth of resistance. Now, another area of antibiotic overuse is in the agricultural community. So approximately 70% of the antibiotics in the world are consumed by animals, and it's generally not to treat sick animals, but as growth promoters, which help the animals to grow faster. So they're not needed, but again, you have this pool of antibiotics that are fostering the growth of resistance. So of that remaining 30% of antibiotics that are used in humans, it's estimated that about 2/3 of those are prescribed unnecessarily. And that's when a patient's feeling sick, they've got a virus or a cold, they go to the doctor, they want to get something, and either the doctor, just to be safe, gives them an antibiotic, or the patient refuses to leave until the doctor has actually prescribed them something. So again, excessive use of antibiotics. So basically, we've had this constant battle in the past between bacteria and antibiotics. And as bacteria have evolved to develop resistance, we've come up with new and improved antibiotics that are able to overcome the resistance. And the problem is now, in the past few decades, we haven't been keeping up with the growth and resistance of the bacteria. So this has led to what's been called an antibiotic void. So the discovery of new antibiotics is not keeping pace with the emergence of the new superbugs. And essentially, there's been a 33-year gap between antibiotics that have been newly discovered and approved and gone to the market. And so this lack of productivity of new classes of antibiotics has allowed the bacteria to become resistant to our existing classes of antibiotics without anything new to come along behind. Now, one thing that's very important to keep in mind is that the drug discovery pipeline, so the length of time it takes to discover a new drug from early research all the way through use in humans, is incredibly long, and there's a lot of attrition along the way. So you start off in early discovery, you've got thousands of potential compounds, and as you go through the different phases of drug development, including once you start testing in humans, there are 3 phases. So phase 1 just looks at safety, phase 2 is looking at initial signs of efficacy, and then phase 3 is the final pivotal clinical trials in thousands of patients. And each step has attrition of anywhere between 20 to 90%. So you're losing possible compounds all the way along the way. And that whole process generally takes 10 to 15 years. So if we don't start discovering new antibiotics now, in 10 to 15 years, when resistance may have dramatically increased, it's going to be too late to come up with new antibiotics. So what's really scary if you look at the current state of affairs. So in 2015, there are over 800 anti-cancer drugs in development compared to only 43 antibiotics. And even more scary, if you look at the development phases of compounds that were in clinical development. So cancer drugs look like this standard funnel-shaped attrition. We've got a lot of compounds in phase 1, and because of the failure rate, you only have a half of that number in phase 2, and about a third of that number in phase 3. If you look at the same year in 2015 for antibiotics, it's almost flat, and in fact it's almost a reverse funnel. So the number of compounds in phase 1 are potentially the number of antibiotics that are going to be able to be approved to be used in humans in 10 years' time. And that pipeline looks exceedingly barren. So we need to be investing into more early stage antibiotic discovery and development. So why is this happening? And honestly, it's largely driven by economics. So if you look at the new drug sales for the first 2 years of revenue from different classes of drugs, things like Lyrica, which is a drug used for epilepsy, neuropathic pain, and anxiety, Januvia, which is used for diabetes, have generated over $1 billion in revenues, compared to two of the best performing antibiotics, which have generated only 50 to 80 million revenues. So for a pharmaceutical company, it costs the same amount of money to develop a new antibiotic as it does to develop a different class of drugs, and yet they can make vastly more money off these other classes of drugs. So it makes sense that they're not investing in antibiotics. And part of that's driven by the amount of money that you're able to charge for an antibiotic. So the most expensive new antibiotics that have just reached the market can charge about $1,000 a day. And normally you take a 1 to 2 week dose of antibiotics. So you're talking about $10,000 to $14,000 per patient for a course of treatment, as opposed to the newest, most effective therapy for cancer, CAR T therapy, has just been approved at a cost of over $400,000 per treatment. And that is socially acceptable, and companies are, providers are willing to pay that amount of money to extend the life of a cancer patient by a couple of years, as opposed to antibiotics are one of the few drugs which actually save a life, and they're not valued anywhere near as much as other types of therapies. So this has led to what's been called a perfect storm. So you have this rise in the number of different types of drug-resistant bacteria, coupled with a decrease in the number of new antibiotics being produced every year or approved every year. And that's largely driven by the exit of almost all major pharmaceutical companies from antibiotic development, from nearly 20 in the 1990s to approximately 4 left today. And again, those are largely driven by the exit economic values, causing the major pharmaceutical companies to exit. So we need to come up with new approaches to try to reinvigorate this antibiotic pipeline. So what do we do? How do we discover new antibiotics? So there are a number of different possible approaches. The most effective approach over the last 30, 40 years is to rationally improve existing classes of antibiotics. And this is why we have 5th or 6th generation beta-lactam antibiotics, so derivatives of the very first antibiotics discovered, the penicillins, which are able to overcome resistance. And we're still able to do that, and that does seem to be potentially the most effective way to generate better and improved antibiotics over the next 10 years. Our own research at the Institute for Molecular Bioscience, we've taken one of the old gram-positive antibiotics, vancomycin, and selectively modified it to target it specifically to gram-positive membranes. And this additional binding mode helps to improve its potency over 100-fold, gives a better pharmacokinetic profile, so it lasts better in the body, and it's able to much more effectively kill gram-positive bacteria. So we've been able to develop this to the stage where it's almost ready for human testing. Now, another possible approach is to rediscover old antibiotics. So during the 1940s and 50s, the golden age of antibiotic discovery, lots of different types of antibiotics were discovered from natural products, and only some of them were developed into drugs because there are so many possibilities at the time. So if you go back in the old literature, you may be able to rediscover some other antibiotics which could now, in the rise of resistance, still retain activity. And this is another approach that we've taken within our group. So in the 1970s, there was a class of antibiotics reported called the octapeptins, which are similar to the current last resort antibiotic for very drug-resistant infections called the polymyxins. And the interesting thing about this report in the 1970s is that it reported that these antibiotics were actually active against polymyxin-resistant bacteria. So we have recently reported the first ever synthesis of this class of compounds and found that, yes, indeed, it has activity against bacteria that have become completely resistant to all antibiotics. So this is a much earlier stage program, and it's still got a ways to go, but it's looking very promising that we've come up with compounds that could be last resort antibiotics and potentially have improved safety over the current last resort antibiotics. Another strategy is to try to repurpose old drugs. So there's some drugs which are now being used for other indications which have been found to have antibiotic activity. Target-based approaches make use of the structural biology and improvements in drug design that have happened over the past couple decades. So that's been quite effective for developing new drugs for human therapies. However, it's been spectacularly unsuccessful at coming up with new drugs that are able to kill bacteria. And that's because while bacteria have very unique targets and we can come up with very potent inhibitors of these targets, the problem is generally you have to get inside the bacteria. And bacteria have evolved many different types of mechanisms to keep compounds out and stop them from killing the bacteria. And finally, we can go back to how antibiotics were originally discovered and try to find them in untested chemical diversity. So one approach is to go back to natural products, which is the source of almost all original antibiotic classes. And there have been a lot of different attempts to reboot natural product discovery by employing genetic techniques, growing bacteria in different types of conditions, trying to compete them against each other so they produce other types of metabolites. And it is looking like it is effective. There have been a couple of high-profile reports in recent years talking about the discovery of new antibiotics from natural product sources. And then the other possible approach is to look at synthetic chemical diversity. So over the past 100 years, chemists have made millions and millions of compounds, and a lot of them have never been tested for their antimicrobial activity. So we've started an initiative called COAD, or the Community for Open Antimicrobial Drug Discovery, which is a global initiative to try seeking new chemical diversity to solve the antibiotic crisis. And we're doing this by targeting academic chemists around the world, getting them to send us compounds, and we provide free screening against 5 different types of bacteria and 2 different types of fungi. And we're able to do this because we've attracted significant funding from the Wellcome Trust and support from our own institution. So we've been running for about 3 years, and the program's been incredibly successful in that we've attracted participation from over 200 different academic groups in 35 countries and received and screened over 200,000 compounds. So we've identified a lot of potential hits. It's still very early stages, and we need to follow up and see if any of these potentially can be advanced as new antibiotics. So we think that COAD represents a missing piece of a puzzle in terms of antibiotic discovery. So you have all these compound collections that have never been tested for antimicrobial activity, and you now have a variety of initiatives which are trying to promote the development of hits into drug-like compounds. So the most recent of these is an initiative called CARB-X, but there's also an AMR center. But there's nothing to bridge these, so how do you find these hits that you can then develop into antibiotics? And this is where we think COAD provides validation, screening, the initial development of data which will support further development of a potential new antibiotic. So apart from trying to discover new antibiotics, there are non-antibiotic approaches which may help to solve this post-antibiotic era crisis. So one approach is to use resistance breakers. So this is combining an old antibiotic, which is no longer effective because of resistance, with another chemical compound which can help overcome the resistance. And the classic example of this is either using beta-lactamase inhibitors in combination with beta-lactam antibiotics. So there's that precedent for this, which has been used in the clinic for a number of years. And there are now quite a few different research groups that are trying to find resistance breakers that work with other classes of antibiotics. Another approach is phage therapy. So phage therapy was developed in Eastern Europe in the 1920s and 30s when they didn't have access to antibiotics. And Eastern Europe is still using phage therapy in some places, but now Western medicine has rediscovered this and is trying to develop under clinically defined conditions with very well characterized material. And there does seem to be a lot of potential, although one of the limitations is that it is quite specific to different types of specific types of bacteria. Another approach, as a preventative approach that's been incredibly successful, has been vaccines. So things like meningococcal And pneumococcal vaccines have been very effective at reducing the incidence or the initial start of an infection. And here's a good indication, certainly, that pharmaceutical companies, if there's money to be made, they will be pursuing antimicrobial strategies, because there are a lot of companies pursuing the development of new vaccines against different types of bacteria. There is quite a lot of research going on now into ways that we can boost our own natural immunity systems to try to fight infections and not have to use antibiotics. And finally, there's a lot of research now going on to do, can we manipulate the natural microbiome or the bacteria that we normally carry in our body to try to use those to help reduce the formation or the ability for pathogenic bacteria to grow. And the classic case of this is in C. difficile infection in the gut, which is caused by a pathogenic bacteria that often grows when the normal flora and fauna has been disturbed by antibiotic treatment, and it gives an opportunity for this pathogen to grow up. And so one non-antibiotic therapy that seems to be quite effective is called fecal transplant. So you're actually transplanting the feces from a healthy individual into the gut of a patient that's got this infection. And there are quite significant cure rates to it, which aren't achievable by treating with antibiotics. So the other approach that we need to do is reduce antibiotic usage to prevent the development of resistance against the antibiotics we still have. So there's been a lot of attention put into stewardship programs, so handling antibiotics more carefully, reducing the unnecessary use. But the one thing in the short term that would have the most dramatic impact on reducing antibiotic prescription would be the ability to develop a rapid diagnostic test. So detecting infections right now essentially relies on technology from 200 years ago, where you have to do a 24-hour culture from your bacterial contaminated sample in order to confirm that there are bacteria present. And ideally, what you'd have would be a rapid diagnostic that you could take your blood sample, extract out the bacteria very quickly, and then do, within a couple hours, a test which allows you to identify the strain, the species, and its resistant profile. And this would allow a doctor at the office to say yes or no, you don't have an infection, and definitively decide whether or not to provide an antibiotic. And if you do need an antibiotic, by knowing what type of bacteria, you can correctly prescribe the most appropriate antibiotic immediately, rather than going through a process of empirically giving different types of therapy. And there's actually a significant prize called the Longitude Prize towards the development of a new diagnostic test that would do exactly that. So I think in summary, there is hope for the future that we will be able to come up with approaches that prevent us entering the post-antibiotic era, but we need to be investing into research now. I'd just like to finish by thanking the co-workers that do the research that's leading to both new antibiotic development and new diagnostics at the University of Queensland.
Now I open the floor for about a little bit less than 20 minutes for any questions that you'd like to address. And please go ahead and you can ask the questions in Arabic as well if need be. You in the back, if someone could please hand him the microphone. Great. Go ahead.
Hi, good afternoon. My name is Mawahib Mohsen. I'm from Zayed University. I'd like to thank Dr. Nabil for his presentation on aging. It is much needed in the Arab world. We shouldn't ignore that taking care of the elderly is also an important part taking care of our community. I know that one of the oldest living communities live in Okinawa in Japan. People over there live on an average of 120 years. What can we do to increase longevity here in the UAE? How can we improve our lifestyles to avoid diabetes, heart disease, and cancer, which are caused by poor choices in sedentary lifestyles?
Can I answer in Arabic?
Of course, تفضل تفضل.
أول شيء
First and foremost, I would like to thank you for your kind question and for this note. As we have previously mentioned, aging of the communities is a major challenge in our Arab region and in all Arab countries. If we look into the studies that have focused on this particular age group, they are purely statistical studies. We are simply calculating, or let's say counting, the number of the elderly citizens who have a specific problem. No specific study show any positive or focus on improving lifestyle and conditions. The secret of longevity, as we have mentioned in the slides, we have the genetic disposition, which up till now we cannot change it, and also the prevention element, the primary prevention or secondary prevention. Prevention from chronic diseases is actually conducted in our communities. So there are campaigns for raising awareness about diabetes, heart diseases, and so on. However, there is a shortage in this awareness, because the problem that the elderly is not just suffering from diabetes or one of the chronic diseases. The problem is that they have a host of these diseases, the least of which are only a minority of them suffer from a single health problem. You don't find an elderly or senior citizen who only has hypertension or has dementia or has some kind of arthritis and some kind of inflammation and so on, or depression because his children are overseas. Not just a single one of them, they have a host of these challenges and diseases. So therefore, prevention should be conducted in a more well-studied manner in our communities. In industrial communities, the basic concept is to have to prevent these diseases and how to prevent falls. How to prevent falls, this is really taken a number of campaigns. There is also an International Day for Avoiding Falls. So we need to highlight some of the challenges and problems that elderly citizens are facing. And we just highlight these problems and these challenges as to be focused only on chronic diseases. No, what we need is just not for longevity, but to improve the conditions under which senior citizens live. We need further studies into this regard. Can we say that the bone diseases affect the quality of life and so on? Yes. And that having those variable elements affect the quality of life? Yes, that's true. And from this perspective comes the concept of global geriatric assessment. This is when we have a senior citizen, the physician need to ask about a number of tracks other than the specific health problem he's complaining from. Because let's give you an example. If I'm giving insulin to someone an elderly citizen who has a problem in the nerve system or who has dementia, for example, if I don't really focus on or ensure that his mental capabilities are fine, how can I ensure that I'm giving insulin in the right manner? So having these multiplicity of specializations, this is the major problem. Another question, please. After the lecture of Dr. Nabil, I started feeling optimistic. We are the young elderly. We like this kind of topics. And this means that having a larger number of senior citizens, this means that we are moving forward. So I believe every aspect has two sides. There is social responsibility in terms of looking after the elderly citizens, and the most important element is that a senior citizen, even when he grows old, he should still feel that he can be productive and can be contributing to the community. And the retirement age, sometimes at 50 years old, people retire. We consider him a teenager actually at the age of 50. So how can we accept that the retirement age should be like 50 or 60? So I'm giving you an example as an individual. My name is, well, actually my age is 78 years old. I'm 78 years old. Still, I still feel that I have the ability to work and to produce. And this gives me the drive to continue my life and to be more productive and to enjoy productivity. When you tell me you should stay home, believe me, life will not get any longer, because one will be living without an objective. So work gives you this objective. When you lose your objective, you lose the meaning of life. I believe that one of the most important elements that we should focus on to look after the elderly is making sure that they have specific objectives, and to ensure that we must not focus on looking after them from the social side or the healthcare side or treatment side. We also need to focus on giving them indeed objectives to them. So I stress once again that the Japanese experience, especially in the field of organs and in the field of academic, social and economic life, it really should have never been that ensure that actually sometimes in Japan they don't allow any people in politics unless they are over 60. So they consider them just young people. So even people below 60 are considered inexperienced young people. So to that extent things are considered there.
يستطيع خبراءنا أطباء.
This topic, if our experts and our physicians, if they would pay attention to it and have future plans that makes the senior citizens' life a productive and happy one, not a depressing one in which they suffer from amnesia and dementia, Alzheimer, and all of those aging diseases. So thanks to Allah, we be say, as the poet said, in paraphrasing of that, the verses are my age. She said my age is over 70, and I said an age is not measured by numbers, and I said I have so much more merits to show. So she asked me about my age. I told her 1,000 lovers. She said, "You're too young. You're still in your 20s." That's a paraphrase of the verses of the poem. I would like to thank you for your very valuable intervention, and I actually second every single word you said. And when we look at you, you are an example of a senior citizen who is successful. That's an example that we should follow. And I also would like to second the idea of having researches done by several stakeholders, since it's not genetics is not just a topic that is related to medicine. It's also related to social sociology and labor as well. And United Kingdom, there is an association established since 30 years. It's called the Assembly of Expertise. So each individual has an experience in a certain field and is retired. He may offer his experiences to this association to the entire world. So he could provide a service to an African country, to an Arab country, and so on and so forth, if we need his expertise in this regard or in this field. So if we think that the senior citizens are a tank of expertise, we need to benefit from their expertise. And one of the recommendations that I hope to be added to this conference is a recommendation about never neglecting the experiences of senior citizens. And that what I want to say is that the human being does not reach the level of wisdom until they are old or one last question. In the name of Allah, first of all, I would like to thank the speakers for the presentation. My question is for Dr. Nabil. In the UAE, thanks to Allah, thanks to our wise leadership, we don't have an elderly home, for example, or we consider it a rest home. So we give, and whoever is taking care of the senior citizen is a person from his own family. So if they need special medical attention, they refer them to the specialized centers. Regarding senior citizens, we incorporate them and have them participate in the social activities, so they actually play a very important role in the social life in a way that help them regain their lives. So they're taking part in social events, and they see their peers, and they see the attention and care from the community given to them, and that makes them feel alive one more time. Another thing we have here in Abu Dhabi is that municipality buildings, they specify specific place for senior citizens. So a senior citizen who the authority for building new buildings, they are providing certain premises or designs that is paid for by the country to have a specific place alongside the building or the house, especially for senior citizens. So I believe that the country is paying a lot of attention to this issue, and we thank the late Sheikh Said, may Allah rest his soul, since he laid the foundation of this principle, and we thank his followers to going and continuing this path. I would like to add one more point. Of course, the family should still be connected and strong, but the elderly citizen should take control of his own path. He does not really need care. He is a provider of care. He can provide services. He could be an active member of the community, and that is why I'm tackling this issue in this specific period. Eventually, I would like the speakers to provide us with a final short note, if you would like to give a final note. One more time, I would like to thank the organizers, and I would like just to
Aging always is an issue, but National Institute of Aging, the NIH, define aging right after birth. When your cells start differentiate, they start age. And أنا مع كبار السن ينبسطوا بحياتهم.
I am all
genetic markers. There is something called telomeres. They shrink with time. This is an aging process. Some people born with aging because of a mutation in a gene, progeria. So aging is a very interesting issue to follow in the future. وشكرا again.
كلمة أخيرة يا دكتور.
I would like to thank everyone, but I would like to highlight one point, which is that there are demographic changes. I call it a demographic revolution. We are living in a real demographic revolution with social variables that we need to pay attention to clearly. Not We should never look away from such issues so that we can handle it positively. Thank you very much. Thank you all for your keen attention, and this is the conclusion of the second session. Thank you for all of the participants of the second session. And by this, we conclude the proceedings of the first day of the 23rd Annual Conference for Future Studies. We all invite you to have lunch with us, and then we meet you all tomorrow by 10 a.m. in the morning. Peace be upon you all. Thank you very much.